Good afternoon, everybody. Thank you for being here. I know it's the final session and it may have been a long day, but great to see the audience and the speakers. I'm Dominik Czaplicki. I work at SANO, Center for Computational Medicine. But my background is more biochemistry, biotechnology, and some pharmacology, so I think I can make it in a session that is mostly focused on pharma and biotech. So from drug discovery and drug development to advanced medicinal products like cell therapies, and also some more or less fancy research models in, for example, 3D cell culturing. Great to see you here, and we are just a few announcements. I'm going to make my best to keep the time, although we are running late already. But so let's aim at 10 minutes per presentation, okay? And I will invite speakers to gradually move down here to the technical team who will assist in equipping you with microphones. You can have this one or a similar one or a wearable microphone if you prefer. Agnieszka Włodarczyk is first to speak about collaborative drug discovery solutions that are the next best thing in collaboration about early phase pharma studies. Maybe not only early phase, but Agnieszka will tell you more. So the floor is yours. So sorry, Alexandra. Yes, I need to fix that.
I'm not an actor.
Yeah.
Polish actor.
Alexandra Włodarczyk on the stage. Thank you.
Thank you. So, good afternoon, everyone. I will try to keep it short, but with the essence. So, here is the thing. I want to talk today about the data silos, about the challenges that every scientist, every researcher, the research organizations challenging every day, and how we at Collaborative Drug Discovery, CDD, help them to organize, work smoothly, and store their important data with our platform, CDD Vault. And this platform, we are constantly developing the new features, and only during the last 6 months, we've developed an explosion of new functionalities, which one of us, deep learning AI module. And the question is how to break the silos of many different types of data. So you can achieve it with our CDD Vault platform to create a central ecosystem. Also, there is a possibility to automate it with the CDD API, application programming interface, to have one single source of truth. So I will tell you briefly about our platform CDD Vault. These are all our capabilities, but I will emphasize on the deep learning AI module and tell you how it can help you in your workflow and what are the benefits of it. So you can register any type of data in the CDD vault, like small molecules presented here on this. Okay, sorry. Presented here on this part of this report table, but you can register every type of entity. So it could be also the amino acid sequences, the cell lines, even reagents, instruments, everything you need to have in one platform, one database. So here you can see the general report table, how it looks like, but it can be fully customizable depending on your needs. And from the report table, which I didn't show you before, but here, you can just directly go to the visualization page. And this is just an example how it can look like. So the aim is like you, all your data registered, uploaded to the database are automatically in the background calculated, and those visualization plots are automatically created. Of course, you can edit them. The graphs and the plots, the curve fits are or editable, and the images are in the publication format and quality. So this is good for any publications, scientific publications. And you may ask, with all those information or your valuable data in the platform, how you can track all of this. So you can achieve it with our module, one of our module, which is a sample inventory module. So you can track any vials, samples, tubes, debit amounts, current amounts, everything you need, and you have it all in one place. Also, some organizations need to describe, need to capture everyday experiments, edit them, upload some files. So you can achieve it with our ELN, electronic lab notebooks. Here we see an example for the chemists, but also we have a biology ELN. And now, because I just very briefly highlighted our platform, I would like to pass on the our deep learning AI module, which was developed in-house by our research informatics team at CDD. So I'm not a developer. I cannot answer any very deep technical questions, but I will try to explain how it works. So basically, this deep learning AI module allows you to do the similarity search by converting a small molecule into chemically rich vector, and then perform this similarity search in the chemically rich space instead of doing the research in the chemical space, which is much more faster. And this search is done in microseconds. So there is no time to see the new, not the new, but the similar molecules. So the similarity search is possible in different collections, like Campbell, Shure-Campbell, CDD public database, and also recently in amine screening collection. It was integrated in the way that is very easy to use, and this deep learning module is available within the platform CDD Vault. As I said, it's very fast and secure in the CDD environment. You can compare your molecule with over 30 million compounds commercially available. So it is very useful because any user can compare their hits with the commercially available similarities. And another application of this deep learning AI module permits to convert numerically only a part of molecule, a fragment, and at the same way do the search, but only for the part, to get basically the bioisosteric suggestions. So I don't know on time. However, I want to be short and highlight the benefits, the tangible benefits that this AI module, deep learning AI module, can be useful for the users. Like, for example, you may want to identify or examine the patent status to see if there is no other worldwide already published to refine your marketing strategy, market strategy, and so on. You can as well want to see in the very early stage of discovery if there is no or there is a side effect or the toxicologically profile to check it before going further in the discovery and development process. Thank you. And of course, I invite you to
a quick request, if I may. Can you just, in one sentence, maybe to give a call to collaboration to the audience? I mean, how can we work with you beyond buying the stuff, right? Because this is a fee-for-service business, right?
Yes, sorry, I haven't incorporated this. So as we are a software vendor, we look for the users. So we collaborate currently with over 650 customers worldwide. Those are CROs, startups, biotechs, pharma, academia. Also recently, the Faculty of Pharmacy of University of Jagiellonian is our new customer. And they are using this AI deep module.
Okay. Yeah, maybe academics can also add something to the system. So staying in the topic of AI in drug discovery, let's move to Adam Pacławski. Thank you, Alexandra. And the topic is Prometheus drug AI system, right?
Yeah, thank you. Okay, so imagine that you are standing in bedside of a child that needs a medication, and actually this child has a problem to swallow a standard tablet. I was in that place a few times. Last time was actually 3 weeks ago with my daughter. Or imagine older person that is struggling with a complex medication regime. I see my father there. Actually, now he's taking just 3 pills a day, but I know that some people are taking 5 or 10. And what if I can say that for all of these patients, we could have a solution? For child, That could be a tablet that will easily disintegrate in mouth and could be swallowed without any stress. For this older person, that could be a single medication that could be formulated in pharmacy, prescribed by medical doctor, and there is just one maybe or two of them, not 10. It simplifies this process. And maybe that's not a surprise, because these solutions actually exist, but still its implementation is limited. And I know that using AI, artificial intelligence, we can at least reduce some barriers, or maybe remove some of them. That's why I called this Prometheus, because this is bringing existing solutions to people. So you may thinking, okay, so how to solve this problem? I work at the Department of Pharmaceutical Technology in biopharmacy, and in our work, we focus on dosage form development. On last slide, actually, of my colleague, we saw there was a dosage form. So what is a dosage form? Basically, it's a form of the drug, so it's not just a molecule itself, it's a whole tablet, syrup, suspension that is usable by patient. So in our department, simply we do not discover molecules, but we change them into usable solutions that patient actually can safely use and effectively. So in our department, we develop solid forms like tablets, but within these tablets, we also develop oral dispersible tablets. So the tablets that basically disintegrate easily in mouth and could be swallowed without stress. That could be a child or person, older person with swallowing problems. We also apply novel technologies like 3D printing techniques to develop wound dressings or advanced drug delivery systems. We also teach students how to compound and design compounding medications, how to test them, and how to make these formulations stable. And this is about sterile and non-sterile. And we also develop empirical models to support this process. And our department exists over 100 years. This year we had anniversary. So it's pretty long time, and we have pretty nice experience. But we also know that we need to improve in the development process. We have to reduce the time needed for that development of dosage form. And this is why over 25 years we are actually developing AI machine learning systems to support this process. Why AI? Why we found it useful? Actually, because of development of a dosage form is pretty complex process. AI actually works better than standard statistical methods. Maybe not always it's necessary, but in some cases, that's the best option, the best solution we have. And actually, you can track our achievements through the last 25 years. The head of the department, Alexander Mendik, started this journey, and actually this time it was against the main trend. So he makes always fun that when he developed a model that had 10 layers and 10,000 neurons, everyone was thinking, okay, listen, it's a too large model, why do you waste resources? Because it took 1 month to develop this model. Now we have models with hundreds of layers, billions of neurons, and we found this approach pretty successful. So a little bit about me. I joined the department in 2010, and mostly through my career, I worked with these predictive models. I started with master thesis, and that was my first success, I think, because I was able to make a model that, based on just composition of the tablet, can predict how the drug will be released from that tablet in lab. And I was able to simulate this process without actually producing this tablet and make this test. So it was purely in silico simulation. Later, I joined a few projects. I started my PhD, and during my PhD, I was developing pulmonary drugs. Also, I supported this process with models, empirical models. And I can say, I think that everyone that is working with AI knows that data is crucial. And because I know both sides, this data generation and data use in AI, I know how crucial is it and how to produce good data for models. And it's not easy. And today, I'm bringing to you 2 projects that I'm now developing. The first one is called GoODT. It's a service for development of oral dispersible tablets. And you actually know what is ODT. So it's a tablet that disperses in mouth, so we can swallow it easily. The global market for these tablets is growing, so it's 15 billion now, and within 10 years should be over 45. But why these tablets are not everywhere? Of course, the development process is costly. So it's just cheaper to develop a standard tablet. If there is no need or huge market for ODTs, companies just don't do that. But if we can reduce the cost of such development, if we can make simulations in silico and reduce the time by 50%, that would be great challenge for patients, because then the ODT tablets wouldn't be maybe more expensive and more accessible. And what actually our GoODT system does, for now the system can predict, based on composition and production method, time of disintegration, which is crucial for registration. In Europe it's below 3 minutes, in US it's below 60 seconds. So at least for now, we can select promising formulations and then go with development faster. What is a value for that project? This project can be implemented in any factory that is producing standard tablets by direct compression. So there is no need for special lines. There is no need for, like in freeze drying, to implement something new. We can do these tablets using a standard compression process. And for now, we are starting with this project in acceleration program, and we will try to pitch this project in January. And how it's connected to this Prometheus? It's make this technology more accessible, make it for user. So what we've done for now, we have a model that works pretty well. This prediction error for this integration time is below 9%, and our square is above 0.8. Of course, could be probably improved. What we are planning ahead, we are planning to develop additional modules to not only support the process in predicting disintegration time, but also stability. As far as we know from companies, that's the problem. So we can combine these 2 things together. I think we will have a pretty nice solution, complex solution for ODT. And of course, we invite you to cooperate with us. Especially for now, we are looking for companies willing to start a pilot program that you could test our system for free. And we would like to get the data, real data of using our system. And of course, if you want to develop this project with us further, like for stability prediction, we are also open for that. The second project is AI drug compounding assistant. And we know that personalized medicine is a huge topic now. We are looking for solution, not that one tablet is fitting all of us, but we are looking for a very specific compositions for every single patient. Of course, it is not possible to do it by companies. So it's made by pharmacists with cooperation with medical doctors. And we are planning to develop a system that will make this process smoother. Because both pharmacists and medical doctors know that to create a good composition requires a lot of knowledge, experience in drug compounding, about stability, incompatibilities. And we know as we have great doctors that can diagnose and provide a good therapy, maybe sometimes they struggle to combine different molecules and excipients together to have a nice outcome. And mostly these prescriptions we have are actually very similar to each one. So we are developing For now, it's large language models, but what I see, maybe small language models would fit it better. And so far, we are able to provide information about basic prescriptions, and the system works correctly. And this is an example that I'm giving my students during compounding classes. I can say that when they start, not everyone is able to correctly prepare that. So if the system works, it means it's better than starting students. And so far, we are looking to develop it for more complex solutions. We are not there yet. So going to the end, I invite you to cooperate with us. For GoDit project, as I said, we are looking for pharmaceutical companies or VC funds that can support this development. And for AI drug compounding assistant, we are looking for pharmacists, compounding suppliers, and healthcare veterinary service providers to make this system working. Okay, so of course, I will be available for you if you want to talk about this project. If you have questions, probably I don't have time, but later I will be happy to answer them.
Thank you so much. So sticking in the topic of tools for drug development, let me ask Michał Jagodziński, Voxel, to the stage. Thank you.
Good afternoon, everyone. My name is Michał Jagodziński from Voxel Radiopharmaceutical Production Center based in Kraków. And today I want to tell you a little bit about how metal radioisotopes can be a tool for developing of new drugs. Let's start with the problem. As we could see at the previous sessions, the drug development is a complex process. It is obvious. But let's think about what could help to make this process more efficient. For sure, it would be perfect to track the disease progression and treatment efficiency in real time. And another useful tool can be that tool that allow us to assess pharmacokinetics and biodistribution of new drug candidate. This above applies to both clinical and preclinical studies. I have a question for all the audience. Who know the positron emission tomography method? Yes, I see few hands raised up, but not much more. So I will a little bit introduce you this method. This technique has been available to patients in Poland for about last 15 years. And it requires to administration to the patient so-called tracer containing radioactive isotope. This isotope should have special properties. It should emit positrons. And then this tracer accumulates in the patient's body at the place of the lesion, and it can be precisely located thanks to a special scanner. This technique uses a phenomenon of annihilation of emitted positron and electrons in the patient body. The use of this technique depends on the tracer. And the most common application of PET are oncology, which use FDG, so the glucose labeled with fluorine 18. For cardiovascular application, we have, for example, rubidium 18-2, and for neurology application, for example, dopamine labeled with fluorine 18. This technique is valued for high sensitivity and resolution, and what is important, it allows to see how drugs behave in real time in the body. Let's give me a few examples how PET could be used in the drug development. The most popular tracer used in human PET is, as I mentioned, is FDG, so the glucose labeled with fluorine 18. This compound accumulates in the place of increased glucose metabolism, which is characteristic for several cancers, thanks to which we can track tumor growth and response to treatment. Another example is prostate-specific membrane antigen labeled with gallium-68 isotope. This compound, so-called radioconjugate, specifically binds to the target protein that is characterized with prostate cancer cells. And this technique can detect even single cells in the patient body in place of cancer and some metastasis, further metastasis. And maybe I will mention also about preclinical studies. PET can be used in small laboratory animals through a method called microPET, and the principle of this method is the same. And the only difference is size of the scanner. This method is especially used for determination of the distribution of the drug. And finally, this is immunopET, which use longer-lived isotopes like zirconium-89. And it can be used to track monoclonal antibodies in the body. In the immunopET method, to the tested antibody, the special chelator is attached, and then this whole compound is labeled with radiometal with appropriate properties. If we need to track longer time the antibody behavior, we can use zirconium-89, which has a 3-day half-life. The other option is copper-64, for example, which have 12-hour half-life. And the classical example of this technique is trastuzumab labeled with zirconium-89, which targets each AR2 receptor. Thanks to this, we can assess each AR2 expression in the metastatic lesions. We get information regarding antibody targeting abilities, and we get distribution profile, for example. And we can monitor this compound in real time. And thanks to this, we get some kind of personalized medicine. And now I want to say a bit about my company, Voxel Radiopharmaceutical Production Center. We are a specialized facility with expertise in production and development of new radiopharmaceuticals. We have infrastructure for research, production, and distribution of radioisotope. Here on the picture, you can see our cyclotron. It is a small medical cyclotron. We have now 2 cyclotrons, one in Kraków and one in Warsaw. And now we recently started to build a new facility in Katowice, and in the near future, we will have the third one. We are marketing authorization holder for our own registered radiopharmaceuticals, and our sites are fully equipped with quality control laboratories and microbiology laboratories. We get all necessary GMP certificates. And what we could do to offer to speed up the clinical and preclinical trials. As a Voxel Group, we have a network of diagnostic centers, and we offer a wide range of highly specialized diagnostic procedures in radiology and nuclear medicine. We possess appropriate infrastructure for research, production, and distribution of radioisotope. Our center could supply a set of radiopharmaceuticals and isotopes such as copper-61, copper-64, gallium-68, zirconium-89, and fluoride-18, and so on. And our R&D laboratory, that is a part of Radiopharmaceutical Production Center, deals with new tracers development, and we have experience in preparing registration documentation, so we can also offer some regulatory support. Our microbiology laboratory performs purity and environmental sample tests for external customer also. We offer access to our cyclone-produced isotopes and radiopharmaceuticals. We have expertise in supporting drug development and regulatory compliance, and we offer broad range of other services. And our facility is located here in Kraków at the 5th Medical Hospital at Wrocławska Street. And now we perform some projects. Now we start to develop Copper-64 radio tracer for examination neuroendocrine tumors. And we start to clinical trial with it. And that's all. Thank you for your attention.
Thanks a lot. I hope there is also some research collaboration opportunities in this publicly listed company that we are proud of, Voxel.
This year we start collaboration with Oncology Center in Gliwice, which use zirconium-89 for tracking a monoclonal antibody, which is used for treatment of glioma, so the brain cancer.
Targeted therapy, so called. Okay, thanks again.
Thank
you. Just if you haven't noticed the speakers, the guys here are to help you to keep the time, so they will give you a notice. And they will give you a notice a minute shorter than previously because we are running late. But Maciej Szaleniec of the Institute of Catalysis and Surface Physicochemistry I'm sure, will keep the time with the very nice topic of enzymatic systems for drug candidates. So the floor is yours, Maciek.
Thank you, Dominik, for introduction. I will keep my time because I have a stopwatch. I will just pick me to stop me talking to you. So I would like to talk about multi-purpose enzymatic system, which is a tool that can be used in drug development and synthesis. And that's our job in Institute of Catalysis and Surface Chemistry, to develop tools for you who struggle to get new drugs to the market, for the old biotech companies doing this. So first of all, I would like to say that we have to switch our industry, which for 150 years is basing on, well, oil, let's be frank, to something more sustainable. And there are two great sources like cellulose and lignin that can be converted to aliphatic acids or aromatic acids. And from these compounds, you can do mostly everything, provided that you know how to reduce it to some compounds which can be then converted to drugs. And enzymatic catalysis is the sustainable way to do this. So this is actually what we are trying to do. And one of these tools is aldehyde oxidoreductase. It's an enzyme we are researching recently for some years. It's a very unusual enzyme because it has a tungsten in its heart, in the active site. That's the heaviest element found in biological molecules. Well, as you can guess from the name, it's oxidizing aldehydes to carboxylic acid, which is basically not very, well, I would say, interesting process. It's used by bacteria to detoxify its metabolism from aldehydes, which are toxic. Here you have the examples of benzaldehyde going to benzoic acid. The enzyme is at the moment a recombinant enzyme, so we can produce it in big quantities. It's overexpressed in a non-standard system, so it's not E. coli, it's Oromataleum evansi. It's our proprietary expression system. You can deploy it in cell-free system, you can deploy it in a whole cell system, and there are a number of electron acceptors you can use in the reaction. So normally it's NAD, which is also relevant for my talk, but also you can use other compounds like DCPIP, methylene blue, benzyl violgens, all the compounds which you can plug into electrochemistry. This is also important. So here in Kraków, we did discover that this enzyme, which normally oxidizes aldehydes, the direction can be actually reversed, and it's a hydrogenase. So it's a first in the world hydrogenase, which is in tungsten active site. Normal hydrogenase is natural iron, nickel. Here is a tungsten, so it can oxidize hydrogen to protons and get these electrons and use them to reduce carboxylic acid to aldehydes. So that's the first use of this multi-tool we are proposing. So you can reduce carboxylic acid to aldehydes with hydrogen, and that's a intrinsically very environmental friendly process, as long as you don't produce hydrogen from oil, right? So if you have used green hydrogen or white hydrogen from some deposits. But there is another use of this enzyme. As I told you, it normally uses NAD to get reoxidized. So you can use hydrogen to make NADH. So this is another use of this enzyme. You can recycle NADH, and this is, as you know, one of the very important molecules in life and also in biotechnology. Well, lots of enzymes, lots of oxidoreductases use NADH to support, as a co-substrate for reaction. NADH is very expensive. So we tested this enzyme, and it's a multi-purpose, it's a generalist. So it's a very broad scope of application. You can oxidize lots of aldehydes, aliphatic, aromatic, heterocyclic, and it's also, you can use this enzyme for detoxification, but also as a biosensor. So you can plug this enzyme to electrode and use this capability that it can react with other dyes that can be then recycled by electrodes to detect aldehydes. And that's one of the applications we are developing right now. But also you can reduce carboxylic acid. As you can see, it's also very general. All these kind of compounds can be reduced. And it gives you plenty of possibilities. So our aim of our project at the moment is to develop IOR as a multi-purpose tool for synthesis of organic molecules. So we can provide oxidation of aldehydes, radioselective, reduction of acid, and an NADH recycling system. And the other objective is biosensor for monitoring aldehydes level in blood and urine. As you know, in diabetes 2, there are some aldehydes produced, and they are markers of oxidative stress in this illness. Of course, they are in very low concentration. You can use LCMS, but not every hospital has LCMS, so it's a very expensive technique. So we think we can actually develop it into monitoring tool or just easy on-the-plate test of these aldehydes. So what we are looking for team members because we already have funding. We only don't have people. So that's actually now a very big problem for all the biotech. At the Institute, of course, we are struggling against fantastic biotech companies here in Kraków, which unfortunately are a bit more well-founded than in Polish Academy of Science. So that's, of course, competition for people, but we are also looking for research teams and companies interested in co-development of these technologies and testing them in their labs. So if you want to make such a transformation, we can try to help you, right? So what can you do with this? Yeah, well, of course, just making aldehydes is not really easy process because it's challenging, because you have to struggle against thermodynamics. You think about it, we oxidize everything. Well, in our metabolism, we oxidize and we gain energy. So if you reduce something, you have to add energy. So it's going again uphill the cascade. But it's sort of like with water. So if you want to pull out from one container water through the tube, you can do this up the hill, provided that something pulls down this water back to the container, which is lower. And this is actually the way you build chemical cascades. For example, you can reduce carboxylic acid to aldehydes, which are higher energy, and then reduce it once again to alcohol. Well, let's make a vanillic alcohol. That's a very nice compound added to food as a part of vanillin as a flavor. But also you can, well, this is a cascade we already made, three steps to just benzyl alcohol. Well, this is nothing interesting, but if you just isolate it, you have jasmine. So this is another compound, which is maybe not a drug, but also a very marketable drug. So you can think about that. You can exchange every aldehydes here, every acid, because we can actually reduce everything with this enzyme. Also, another possibilities, we can, instead of alcohol dehydrogenase, we can use aldolases. So then you can make CC bonds, so polyhydroxy acid, or here an example of leading to quinolinic acid, which is a very common motif in many drugs. So, of course, there are many more. I will not bore you with the chemistry. Only thing that just show you another possibility. You can also reduce NADH, NAD to NADH with hydrogen. So you can use it as a very, very efficient recycling system of NADH. And then every enzyme which uses NADH well, whatever you mean, amines, chiral alcohols, chiral amines, you can produce it with this enzyme. So this is a very efficient system for this, especially that you can, instead of hydrogen, you can use green electricity. Just you add mediator, and you can plug this to the electrode. Actually, this system was developed, the first case for making the electricity connection with ATP production. That was done in Marburg University, not Marburg University, but the Max Planck Institute in Marburg. They used 3 enzyme system to make ATP from electricity as a first kind of a bio-ATP electrical system. So, cooperation offer. We definitely are the team building up stage. We are looking for undergraduate student. We have a position for undergraduate student. PhD recruitment, we are just running a kind of an unusual winter recruitment here for the project. So if there is any PhD candidates here in the hall, please approach me. We have postdoc positions, and we are working in very wide international team at the moment: Poland, Netherlands, Germany, and Canada. But still, some new countries are prospectively joining this team. And we also invite you to co-develop this and test in your companies, in your laboratories, as enzyme tool for organic synthesis. Also, we are going to develop as a biosensor for aldehydes in body fluids. And well, if you just want to do NADH recycling, we also can provide, you can test if it works in your system. So thank you very much for your attention, and I invite you to LifeScience Open Space platform, where you will also find this, not only me as person, but you will also find this, well, discovery as a cooperation offer. Thank you very much. And I still have 16 seconds.
Yeah, great. Thank you for the excellent time management. So from a rather small tools for drug discovery, we are moving to something slightly larger. And Artur Biela from the Jagiellonian University here works at something that is perhaps the biggest and the most expensive research tool in this country, Solaris Research Center. And the slightly smaller part of it is the cryo-EM. So the floor is yours.
Thank you. Welcome, everyone. I'm definitely going to keep the time, because it's only one single tool that I want to show you here and present as an option for your research and development of your projects. So first of all, if you don't know where we are located, this is the university campus here, and the Solaris Center is like 800 meters across the field. So if you're not afraid of wild boars, you can go across the field. If you're afraid of them, then you have a little, slightly longer walk next to the tram tracks. It's only one tram stop away. At our cryo-EM facility, we have 2 electron microscopes that can work in transmission mode. Both are in different acceleration voltage settings. So we have 200 kV acceleration voltage for one microscope. This is our screening microscope, and everyone can access it. And the second one is 300 kV accelerating voltage microscope, which is the high-end microscope we are using for our research. Is also accessible for now only for academia, but from the next year on, we will also open it for industrial users. So the techniques that we are covering with those microscopes is the cryo-EM SPA, which is single particle acquisition, the cryo-ET, which is the electron tomography in cryo conditions, and on the glacious microscope, so the smaller one, the one on the right, we have also micro-ED, which means that we can make the diffraction of electron beam on the microcrystals. But going through the details of the method, how we image our samples, first we need to freeze it. We need to freeze it in vitreous ice because the crystalline ice is intransparent for electrons. So we freeze our sample by plunging it in liquid ethane. So we produce very thin layer of vitreous ice with the particles trapped inside the ice. Then we illuminate it with the electron beam, and we record the image of the shades that our particles embedded in the vitreous eyes project onto the detector surface. And then we analyze those images, we fish out the particle shades from the whole images, we cut them out, and we try to align all of those particles one onto another to average those and increase the signal-to-noise ratio. Once we have the averages, which are marked here in the panel C, we can do the back projection of those images and reconstruct the 3D volume. Once we have the 3D volume reconstructed, then we can refine it and we can tell how your protein, how your complex looks like. The second one is the cryo-electron tomography, which works in a very similar way, but instead of recording numerous particles in different orientations, we image only one particle, but we tilt the stage so we know the rotation angles, so we know the relation between the images that we have recorded and the angle between them, so the reprojection is faster, but the quality is a little bit lower because of the electron damage that we introduce into our sample. So just to show off a little bit how this is important for collaboration and for biomedical projects, I would say. I hope the movie works. Yes, this is the structure of the DHS complex with EAF5 alpha protein, which is responsible for polyimine metabolism, which was connected to some neurodegenerative disease. So by combining cryo-EM and the room temperature crystallography, we were able to decipher the full mechanism of amine group transfer from spermidine to the protein. This actually was a base of the team grant for Dr. Przemysław Grudnik, who was awarded with the grant several months ago. The second very important work that was done on our microscope as well was solving the structure of the cytochrome B6F with plastocyanin, and that was awarded with the Parnas Award for the best biochemical publication from Polish group in 2023. And the last but not least is the elongator complex structure. This complex is responsible for modification of tRNA, and by this it controls the efficiency of the protein synthesis. And this particular structure was awarded the FNP grant this year, which will be, the gala will be held next week in Warsaw. And as a part of the team that was involved in this discovery, we are all going with Sebastian Glad to Warsaw to get the prize with him. And if you are wondering how you can access the microscope, that's the most simple way to get to Solaris. There are some turns on the way, but don't worry, they're very easy to overcome. Once you get at least to the point number 3, it's easy. But setting up your account in the system, it's very intuitive. And once you get there, you can apply for time. For academic researchers, if you have initial data and you write a so-called grant application or proposal application, it will be evaluated by the international committee. It will be granted a score and then be given a time on the Cryos microscope so you can perform your experiment and enhance your data in your projects. And of course, the open calls are twice a year. So one is the spring call, which is in March. The second one is in September, in autumn. We have also the rapid access proposals open, which is for the proof of concept experiments, or if reviewer of your paper will tell you, do the cryo-EM, then we're for it. And of course, for industrial users, there is a paid access where you can just call us, apply, and we will give you the estimate of the cost. We will sign the agreement, and then you will get the time. So in case of any further questions, you can find me here or somewhere around the venue. Thank you.
Thank you very well. That was yet another tool for drug discovery, a pretty sophisticated one. But let's move back to the enzymatic methods for a while. Institute of Catalysis again. Agnieszka Wojtkiewicz on the stage.
Thank you very much. Hello, everyone. So I would like to present you the project that is called Tribioster A in English would be maybe, and it's about bioproduction of different triterpenes as an alternative for healthier, an alternative for steroids. My name is Agnieszka Wojtkiewicz, and I'm from Institute of Catalysis and Surface Chemistry, Polish Academy of Sciences in Kraków. So today I would like to tell you a little bit about pentacyclic triterpenes, why the bioproduction is important here, a few applications, and what is our offer and our needs. So pentacyclic triterpenes are sourced from nature. They are produced mostly in plants, in bark, leaves, and fruits, and they serve as secondary metabolites with protective roles. We can obtain them by natural extraction from plants, and it can be supported by chemical synthesis and biotechnological methods, which are here more and more popular. And some applications of those compounds, they can be considered as anti-cancer drugs. They have fantastic anti-inflammatory properties. With some modification of the structure, with some decorations, they can be considered as fantastic antiviral compounds as well. They have antioxidant activities, and they can, in general, serve as an alternative for steroidal drugs. So what is the impact on the society and environment by using those compounds? It supports sustainability, it advances green chemistry, and offers innovative solutions for health, cosmetic, and even agriculture. So this is the structure of one of those compounds, and they are quite available in nature. For example, lupeol is extracted in 20 grams per each kilogram of dry matter. They have documented applications with nice in vitro, preclinical, and even clinical trials. The bioavailability is known, and they are safe. So here are some examples. For example, betulin and betulinic acid are used in supplements and in a drug. There is a phase 3 clinical trials for oleanoic acid in oleogel S10 with a very nice result that is shown here. The wound healing, which is better, is the place where the ointment was added. It's after 2 weeks. So this works pretty well, and it's under FDA regulation right now in the office. Another example is using of oleanoic acid and ursolic acid as oral administration that is already in phase 2, which is ongoing. So whenever we think about some comparison that is already in the market, that would be steroids, which are essential, very potent, versatile. However, they have side effects, which we know because they are so similar to our hormones. Nevertheless, there is a huge demand on the market. In 5 years, there is a prediction that the market will double. So some alternative would be nice to have. And whenever we think about steroidal drugs, there is one tiny modification of a structure that is shown here, extra double bond in the structure. And this is, as a result of this small modification, we have a nicer anti-inflammatory potential. It's 5 times more potent. Then we can reduce the dose of such compound. So is there a chance to do the same for pentacycliterpenes? And the answer is yes. We can also make more active pentacycliterpenes, and they are also found in nature. However, they are produced in a small amount in plants. It's not 20 grams, it's milligrams. And they can be found, what is interesting, in plants that are known from traditional medicine, for example, Chinese medicine. And in our institute, we developed a bioproduction of triterpenes to active form with a nice conversion rate. And with this method, we can right now gain or receive or produce something like 5 grams of such a compound, which is 1,000 times better than in nature. And what is great about this bioproduction, we can scale it from gram to kilograms and to 100 grams. So we have nice bioproduction to produce more active derivatives. That bioproduction is efficient and stable. And what is also important is the rapid manufacturing timeline of it. And some applications, we could use it as supplements due to their anti-inflammatory effects and also synergic effects with antibiotics. They are nice as multifunction cream agents because of rejuvenating activity and, again, anti-inflammatory activity. We could use them in wound healing with this reduction of dosage in such ointments. And they can be considered as drugs in such diseases as diabetes and neurodegenerative diseases, Alzheimer and Parkinson. So we offer innovative solution that supports sustainability, green chemistry, and it's near implementation stage. Right now it's TRL 5 to 7, and it's IP protected already. And this method is a nice alternative for steroidal drugs, mostly because of its anti-inflammatory properties, with less side effects, that is safe, and there is a huge market demand for such solutions. And what we offer, we have novel compounds in gram scale right now. We have ideas for applications. We have nice scientific collaborations to test new applications and to prepare new formulations. And we have experience in patenting and industrial implementation. What we need, on the other hand, is, of course, funding for that project. But here, mostly to build a team and to run studies and develop the project, because we have quite nice facility to run this project. And then what we also need is someone with experience in regulations of medical compounds, and someone that is who's open to advanced technology, including clinical trials, and maybe something else that you can provide us and we don't know about yet. And I would like to acknowledge here my institute that supports this project financially, and Tomasz Więcaszek, Natchem company, who supplied us with pentacycliterpenes. If you have any question or something to discuss, please contact me. Thank you.
Thanks a lot. Thank you for the excellent time management again. The Institute must be famous for that. So now Selvita and yes, Maciej Olszewski, the floor is yours. And this will be a slightly different tool for drug discovery, which is an in vitro system, right?
Yes, this is.
So please go on. Thank you.
So thank you for coming. I represent Selvita, which is a quite unique company in Poland because we are a strictly CRO, contract research organization with a global reach. So we work with clients from all over and we face global competition. So we have to also keep up with the competition. So we develop new methods constantly to address the needs of our clients. And this particular example that I'm going to present today is an example of imaging-based platform, so a microscopy, but taken to the next level in terms of quality and quantity, let's say. So our new research center opened March last year. We moved over to a new building, and this is where our imaging platform operates. There is maybe two major suppliers of this type of imagers. PerkinElmer is one of them. The equipment I'm talking about is an automated microscope that can work 24/7 with a robotic plate feeder, and it can generate anywhere between 1 to 2 terabytes of data per day. So this creates the whole new set of challenges on how to store, manage, and analyze this amount of data, image data, because there is no point generating the data faster than you can analyze it, right? So we faced the problem. Lack of fully automated system capable of image acquisition, and then some multi-parametric phenotypic analysis. What does it mean, multi-parametric? When you have an assay which is simple, so there is more or less of a certain protein, or it is located in the cytoplasm or in the nucleus, it is very easy to create an assay to measure that phenotype. But if your phenotype is a change of shape, or some coordinated changes in few parameters, then it's much more difficult to quantify it. And in the end, we need to quantify our changes, our phenotypes. So this is an ideal area to implement some computer vision and advanced image analysis solutions. So for that, we worked with Artigen, our sister company that specializes right in that. And we've created some common approach to those challenges. We decided to start with the two important therapeutic applications, which is neuroinflammation, which lies at the root of many neurodegenerative diseases, including Alzheimer's disease, and fibrosis. So this is the process in which tissues get scarred and are no longer functional. And one example is idiopathic pulmonary fibrosis. It's a very serious disease for which the reason is not really known, but it is known that the lungs get scarred, and it would be ideal if we could stop this process. So what we created was a platform that has an imager running at Solvita, a data link that transfers the data to Artigen, automated quality control algorithms that identify the images that may not be perfect, try and improve the quality, and then apply some advanced computer vision models to analyze those images. And there are several ways of analysis. When the required analysis is relatively simple, we can do it in our software, Harmony. We can also create some relatively simple human-supervised AI models where we, as humans, train this algorithm and at the beginning indicate the cells that are normal and changed, and the algorithm will extract from the set of 50 or 100 features, so measurements per cell, including size, shape, intensity, granularity. The algorithm will identify the ones that are important and then recognize those features in new cells. And then there are fully automated models, self-learning models, based on neural networks rather than linear equations. And those are used for the most challenging phenotypes. And by challenging, you mean the ones that you, with your human eyes, can actually see this. But you cannot put a number on it. So one example is the pathway for LPS, which is a bacterial component that induces inflammation in cells. And this inflammation, for example, one of the phenotypes is what you can see here. The cells change shape. This is a microglia from the brain, and you can see the cells change shape, not really size. And you want to be able to quantify this process and find the parameters, hopefully automatically in this, that describe this change. So what we have done, this is our in-house Solvita machine learning algorithm. We have shown some images of treated and non-treated cells to the software, and software will try to extract the features that change most. And from this, you can see we have activation, So with LPS, we induce the inflammation, we get the activation, and then on the bottom panel, we have a compound that prevents inflammation, and it will reverse the phenotype. So now we created a certain assay that can recognize anti-inflammatory compounds. That was the purpose. But in our hands, it required 24-hour treatment. So we were wondering, can we have a more sensitive assay or data processing? And we've given the images to Ardigen. And they looked at both nuclei and the cell shape. So we have a various fluorescence channels, and we can see this cell mask. This is a cell shape, Hurst nuclei, so this is nuclei. And amazingly enough, this algorithm can actually see and quantify the changes after 2 hours of exposure already. 2/6, we can see it after 24 hours. So only looking at the nuclei of cells after 2 hours of exposure to inflammatory component, this algorithm can see the difference. So this is amazing, and the actual values of EC50 are very similar to ours after 24. And you can see the progression is expected. Short exposure requires higher concentration, but the sensitivity is really great. So that's one example. The other is a fibrosis, which is a scarification of tissue. This is where fibroblasts convert to myofibroblasts, and one of the easy phenotypes is a higher expression of smooth muscle actin. This is a certain protein. And you can see this is the easy phenotype. It's dark and then it's bright. But also you can approach that by looking at, again, the shape of the cells. So on the left you can see healthy cells. They are long, they are narrow, spindle-like, and when they are exposed to TGF-beta, something that causes fibrosis, you can see the shape changes. So now we can quantify the two, and this is our simple in-house AI at Cellvita, and we can quantify it. Please keep the number 185 in mind. And then if we now look at those same cells through Ardigen's eyes, and again through nuclei and cell shape and 3 parameters together, smooth muscle actin, cell mask, nuclei, now we get some numbers, and amazingly again we get a very similar EC50 values than we got from our algorithms. So this is 0.25. We had 0.18, so it's very close. And this is self-learning algorithm, so we don't need to train it. It will recognize those changes itself. So what we were able to create, and this is just 2 models, but this platform is open, right? So we now have a working, a functional workflow where we can image, transfer, quality control, and analyze the images between the 2 partners of this consortium. Importantly also, algorithms can access the proprietary data format that PerkinElmer created. So we can now go open source. While Harmony is a closed system by a provider, and there is no way to enhance its functionality. So we have that. Economical value. If we just look at the shape of the cells, we don't need to stain them with antibodies. So we conserve reagents, we conserve environment too, human work. We can be more competitive on prices because we extract more information with less work and reagent expense. So for now, the platform is ready for business in those applications that were developed under the program. If a new analysis is required, then we can quickly create the assay in the lab and then create a model to analyze it. And then if anyone has a great breakthrough ideas for computer vision and image analysis, we are now able to incorporate that because now we have an open source interface and we are always looking to enhance these functionalities. So these are the types of maybe collaborations that we might be looking for or services that we provide. Thank you.
Thanks a lot. Now let's move from bioassays to biomaterials. And this is again the Institute of Catalysis, but yet another research group. And drug delivery systems here on the stage with Magdalena Goncerz.
Thank you so much. Hello everyone, my name is Magdalena Goncerz, and today I would like to say something about one of the topic of our research, which is dendrimer platform for drug delivery systems. So as most of you know, the cancer cases are still growing, and the cancer is still one of the main reasons of the mortality around the world. And the most common method for cancer treatment is chemotherapy, which of course is very effective, but also is characterized by a lot of limitations, such as failure to use the full therapeutic potential of the drugs because of their low biodistributions. Anti-cancer drugs are also characterized by lack of selectivity, which means that they are toxic not only towards the cancer cells, but also normal cells. And that's why the anti-cancer substances are considered as high toxic. Cancer cells have also ability to become the drug resistance. Anti-cancer substances are also characterized by short half-life, and that's why they are rapidly eliminated from the body. And in general, chemotherapy causes the many side effects during the therapy. So our question is if we can deliver drugs more precisely and efficiently for improving the quality of the chemotherapy. And our proposal is to use PAMAM dendrimers as drug delivery systems for anti-cancer substances. Dendrimers are polymer symmetric nanoparticles with regular and well-known structure. When it comes to dendrimer structure, we can divide it into main parts. There is the core inside the structure, the branches emanating from the core, and the surface layer with many functional groups. And properties such as precise structure, multifunctionality, controlled size at nanometer scales from 1 to 15 nanometers. As you can see, spherical shape, also monodispersity, high stability in water solutions, and lack of toxicity makes the dendrimer special candidates for drug delivery systems. And because of the unique structure of the dendrimers, then they can interact with the drugs in two different ways. So they can encapsulate the drug particles in the internal spaces and also immobilize the drug particles on the dendrimer structure due to the presence of many functional groups. And dendrimer and drugs can create very stable complexes which can be used in the biological conditions, where the drug can be released in the controlled manner into the cell matrix, ensuring the increasing of the selectivity of the drug and reducing its toxicity. And that's why the dendrimer drug formulations are the object of many research. And dendrimers have been studied for use as nanocarriers for drugs such as paclitaxel, cisplatin, gemcitabine, curcumin, docetaxel, or cabazitaxel. And some of the dendrimer drug formulations are currently the object of the clinical trials. And when it comes to our achievements in dendrimer-based nanocarriers, we are highly experienced in detailed physicochemical characterization of the dendrimer and drugs. We also have very good methodology to optimize the conditions of the dendrimer interactions with anti-cancer drugs, such as, for example, 5-fluorouracil or doxorubicin, which are drugs which are very common used in the medical practice, but they are also characterized by a lot of limitations, such as drug resistance or cardiotoxicity. In our studies, we proved the selective activity of the dendrimer complexes with the drugs in vitro conditions. We also observed a different intracellular distribution of the complexes in the cancer and normal cells. And also we observed, for example, in the case of doxorubicin, pH-dependent release from the dendrimer structure. And when it comes to the collaboration we offer the functionalization of the dendrimers with active substances for creating highly targeted and efficient nanocarriers. And we are performing our research in few main steps. we are performing verification of the encapsulations level of the drug in the nanosystems. We have tools to determining essential physicochemical parameters of the created nanosystems. We are able also to monitor the interactions between the nanosystem and the different surfaces, such as negatively charged metal surfaces or lipid bilayers. And also at the end, due to the collaboration with the medical department, we are performing in vitro assays for verification of the biological activity of the nanosystems. And according to the recent data, about 100 nanomedicines are commercially marketed, and about 500 of them are under clinical trials, which demonstrate the potential of the nanoparticles in clinical use, including cancer treatment. And as you can see in the graph, the nanomedicine market will grow over the next 8 years. And when it comes to the dendrimers, it's worth to say that they can be not used only for drug delivery systems for medicine and pharmacy applications, but also for imaging tools or biosensors. And we have also tools for designing the biosensors for biomedical applications based on the dendrimer. And when it comes to the collaboration, we are looking for academic or the R&D partners which are interested in our study and can help us to develop our dendrimer-based nanosystems for higher levels. So, for example, for in vivo studies or clinical implementation. And from our side, we also offer to use our experience in functionalization of the dendrimer or different nanoparticles by selected or the new drugs. And if you are interested in collaboration with us and interested in our study, I am inviting you to visit our webpage, where you can find more information about our publications, projects, achievements, and our equipment. Thank you so much.
Thanks a lot. And staying in the area of biomaterials, now in more like mucoskeletal applications, so to say, let me invite Karina Niziołek from the Kraków University of Technology. Thank you.
Hello, everyone, once again. And I'm saying once again because you may remember me from the previous session, MedTech session. I already had my presentation. My name is Dagmara Słota. This talk should be done by Karina Niziołek. Unfortunately, due to personal reasons, she couldn't attend this event today. That's why I have a pleasure to present our research. We are working in the same group, so that's why I can also present her speech. So the topic of the presentation is hierarchical biomaterials as a solution to the problem of treating osteochondral defects. And let me start with a short introduction about the problem. So the problem that we are thinking here in this project is osteoporosis. Osteoporosis is a progressive disease that is characterized by weakening of bone. So basically, bones becoming more likely to fracture. This disease affects millions of people worldwide, particularly aging populations. Also, it is a really big challenge for global healthcare to handle osteoporosis, because the fact is, once you have osteoporosis, you cannot really stop it. Yes, so it's a progressive disease. Everything you can do is slowing down the progress of osteoporosis, but you cannot just stop it. So that's why it's a really big problem. And also about osteoporosis, it's affecting many people worldwide, millions of people worldwide. However, mostly female, because hormonal economy is related with osteoporosis, and it's changing after menopause. So that's why female are more often suffering with osteoporosis. And also I'm saying suffering because having osteoporosis means that patients feel constant pain and also have a problem with the movement. That's why it's also a challenge for us, as for researchers. So we ask ourselves if we can do something with this disease, if maybe there are some reasons, some biomaterials that we can project to help those patients that's suffering with osteoporosis. And we had a short, it's not that short project, it's 3 years project, but yeah, we had a project that helped us obtain a material within we can fight with osteoporosis. The aim of the project is to develop a multifunctional ceramic polymer biomaterial that combines mechanical properties with therapeutic functions while adhering the green chemistry principles. And I will try to describe for you this project without using super scientific words, because I know that not all of us are related with biomaterials and polymeric and ceramic science. So anyway, the first step in our project was to obtain appropriate ceramic phase. We obtained 2 kinds of ceramic powders and bone cements. Later we used only ceramic powders, but with bone cement the interesting thing is that we can just use bone cement to fill bone defect, which is also really interesting. However, focusing on the ceramic powders, it's not that new to use ceramic powders, but what we have done is to modify those ceramic powders with metal ions. So we modify ceramic powders with magnesium, manganese, and zinc, and this way we provided antimicrobial properties for those powders. And then the next step was to combine those ceramic powders with metal ions to polymeric phase. And this way we obtain hierarchical structures, gradient biomaterials that exhibited antimicrobial properties, also osteoconductive properties, but what was really important for us, appropriate mechanical properties. And you may ask, what does it mean, appropriate mechanical properties? And it's really interesting and really important in case of osteoporosis, because the tissue that we would like to replace in the framework of fighting with osteoporosis is osteochondral tissue. And it's a really specific tissue because on the one side it's solid, it's hard like a bone, and then it slowly turns to more like a sponge, like a cartilage tissue. So it's really hard and solid on one side and really elastic on the other side. And that was the challenge to obtain this kind of material. However, we handle it, we obtain this kind of gradient structure. And what is also really important, we obtained this material in an eco-friendly and waste-free technology using photo-cross-linking technique. And this technology meets green chemistry expectation because we do not generate any waste, any byproducts with this technology. And because it's a scientific project and we basically started 2 years ago, we are still have many research that we would like to perform to check our biomaterials in the best way that possible. So we are looking for collaboration with research institutions, but also with clinical organization and industry as well. We believe that together we can drive innovation that will transform global healthcare. And we can provide you, of course, with early access to innovative technology. Of course, no research would be possible without adequate funding. In this case, it was a project from National Science Center, Hierarchical Approaches for Osteochondral Tissue Engineering. And here on the slide, you can see our very feminine team. Yeah, and that's all. Thank you for your attention. If you have any questions, feel free to ask here or through LifeScience Open Space platform. You may ask me, but also Karina Niziołek that should be here instead of me. So thank you.
Thank you so much, Dagmara. And right now we had already at this stage a US-based company, some researchers from notable Krakow's universities and Polish Academy of Sciences, publicly listed companies like Voxel and Cellvita, and finally we have an SME, originally from Kraków, with a little help from the Jagiellonian University. Marcin, the floor is yours. Real research and 3D in vitro cell cultures.
Thank you very much, Dominic. So I will try to be brief, although I have quite a few slides. So please stay with me. I know it's late, so please focus for the remaining 10 minutes. So what we do, we basically make hydrogels for 3D cell cultures, and I would like to introduce you to some breakthrough discoveries we've got for the last year. So probably many of us know that there is a huge problem in drug discovery, mainly that whenever you go through the initial process of the preclinical stages and you get to the clinical phase, there is basically like 97% failure rate in oncology, which is, when you take into account the cost, which is actually totally unacceptable. And there are basically two main reasons. One is false negatives, the other one is false positives. Basically, companies invest in drugs that do not work and they close programs that could actually work. And that's a really huge problem. So I will show you two case studies of how we can really improve that. And I will first focus on the pancreatic cancer. So on this slide, you can see gene expression profiles. And you can see that when you do the 2D cell culture, which is on the right, the gene expression profile is quite heavily shifted towards, I would say, non-physiological one. And then when you look at the two types of 3D cell culture, which is in this case young and old, you can easily see that this is much closer to in vivo. And this is actually what we will build on. And obviously that's also visible when you look at the morphology of those cultures. In our case, what we do, we basically make the hydrogel, we use it to seed the cells on top of the gel. And because of this very simple reason, we actually get the growth of the three-dimensional tumors on the gel, not inside the gel. And this is actually crucial when you take into account that the real physiological tumors, as you can see on this immunohistochemical slide there, they actually are on the tissue. They are very rarely inside the tissue. You can make a cross-section that it looks like it's in the tissue, but they're really on the tissue. So what we actually offer are products, but we also do services, but the products are really the primary goal of the company. And not to take it from my words, you can actually see that that what we grow is actually 3D, and it is actually nicely, it can be visualized, it is actually on the gel again, and you can see that it's a nice whole 3D structure. In a second, we will go through the whole video, and this is not an animation. This is actually a full visualization of a large, like 400 micrometers, three-dimensional structure. Just give me. And by the way, this was also stained not only with like biochemical stains, but also with antibodies, which will be important in a second. Just give it a moment. We will go into. Oh yeah. So now we can see that you could see that there are cells inside, and because of that, it's a really dense three-dimensional structures. And for those of you who worked with pancreatic cancer, you know that that's actually again closer to what you can find in patients. So what is really the challenge? And I will focus on the pancreatic cancer case study. So really the problem is that there is close to no treatment, and this no treatment comes from those two problems I already mentioned, which are false positive and false negative results that you can get. So if you would test gemcitabine on 2D cell culture, it works marvelously, whereas when you do it, when they did phase 2 and phase 3 studies, they actually improved the partial response from 5.4 to 12%, which, let's be honest, is not giving any hope to anybody. The same goes for 5FU, especially in combination with gemcitabine. Yes, it improves it a little bit, but when there is a sick person, an improvement of several months with a lot of suffering is not what you're looking for. And what I'm going to show you are the most standard techniques and how they fail. So this is a study where they tested, the whole study tested like 20 different cell lines. Here we have just 9 of them. And you can see that for the classic drugs, again, like gemcitabine or 5FU, all of them work. All of them are just wonderful. And you could say, well, this is the outdated 2D cell culture model. So let's move to the most advanced ones. There is a nice study done on, I don't remember exactly, it's like 30, 40 different patient-derived organoids from pancreatic cancer, and all of them showed exactly the same thing. All of them showed that gemcitabine, 5-FU, oxaliplatin, and SN38 actually worked. So these are the classic chemotherapeutic drugs, and that's actually one of the main problems, because they went through the whole process and they do not work. So here I really wanted to show you what we have discovered throughout the last year and what is, in our view, one of the biggest hopes, especially for pancreatic cancer, but we see very similar things in glioblastoma, and we're expanding this to many other types of cancer. So in the 2D, can I do this? No. Yeah, I do. Yeah, no, that's not going to work. Okay, so when you look at here you can see just a comparison of 2D cell culture versus our 3D cell culture. The 2D is the red line that goes down. This means that the drug basically causes the cell death. It's actually GR, so growth rate inhibition. And you can see that those drugs, 5-FU, gemcitabine, SN38, and oxaliplatin, they actually, it looked like as if they worked. And this is what I showed you in the 2 previous slides. But when you try to test them on our 3D structures, one of those actually I showed you in this big image, they basically do not work. So when you take 5-FU and PANC1 is a pancreatic cancer cell line, even increasing the concentration by like 1,000 times does nothing. The same goes for oxaliplatin and SN38. And then if you look at gemcitabine, so in the left down corner, you can see this very slight change. And this slight change is actually very much aligned with the clinical data that we're showing, increased from like 2% to maybe 5%, which is, it's something, but it's nothing really valuable. And you could easily say that, sorry. You could easily say that this effect just comes from the fact that it's a large structure and it's not dividing. So both of them are false. So the fact that cells are dividing, you can see here on the right-hand side, the upper graph, we actually see that the cells are growing. And we also know that the diffusion through the whole structure takes about 4 hours. So the 72 hours exposure to drugs is much more than the time needed for the drug to diffuse in and out of the structure. And this, by the way, this is, by the way, one of the things that we're looking for a collaboration to investigate. We have done full gene expression profile and full proteomics data on those models. We already have a hint why this is happening, but we are very open to collaboration in this field. So the advantage of what we do is coming from several points, but we didn't establish exactly why we observed this, but we've repeated this many, many times. One of the hints is most probably that what we offer, what our gels do, that we can actually test different stiffnesses and then actually expect different drug responses. And let me, since the time is running, I will just jump a little bit. So the other thing I wanted to add, so everything I was talking about so far was focused on small molecules. But honestly, one of the biggest advantages we can offer, because of the surface location of that 3-dimensional tumor, is the fact that you can test very easily large molecules, biologic, ADCs, any type of antibodies, anything that is big enough that will not diffuse through the gel. And this is something you can see here on the down left corner. So this is every of our competitors. The diffusion through any of the other competitive hydrogels is very, very hard, because it's like one of our clients told me that they need 8 4-hour washings to do this. In our case, there is no problem because they are on the surface, so you can very easily test biologics, any biologics from, again, from antibodies to oncolytic viruses. And don't take it for my word, because we've done a case study, and here on the bottom you can see treatment with without immune cells. So it's the bottom line of picture is a control, and the upper one is an actual experiment. So with CTLA-4, it was a negative control for a mock antibody that didn't work, and then PD-1 and anti-Vista were actually the 2 ones that we tested. From the pictures, if we would go closer, you can see that the 3 structures are actually, they are disrupted, but it's not that you have to look at the pictures, you actually can measure the viability. And here, we were able to measure, and by the way, the atezolizumab is anti-PD-1, so you can see a decrease in cell viability by about 20%. But what is actually even more important is that that 20% decrease in viability happens within about 5 days of the treatment, whereas the half-life of the antibody in the human body is about 2 weeks. So what we see is that 20% decrease, and in patients you can see about 40% decrease in comparison to chemotherapy. which is again much more physiological, because if you do the same test in 2D cell culture, you're going to see like 98% decrease in cell viability because of the simple access to every single cells. Therefore, again, we're showing a much more physiological model. Just to summarize, we're looking for collaborations, especially to learn why we are observing such a high level of resistance, but also to develop more models for immuno-oncology and expand the number of applications and the number of case studies. And we want to apply those models to early stage drug discovery processes. And although we can add a lot in the small molecules field, we also see that there is actually a huge gap in the value chain for large molecules, where it's actually very hard to test any high throughput systems in the complex models because of that simple access of biologics. So we believe we can add a huge advantage there. So I have a few other slides, but I will stop here. So thank you very much for your attention. And if you have any questions, I'm very happy to speak with you later. Thank you.
Thank you so much. Thanks a lot, Marcin. So now we stay, so 3 presentations more to go. Stay with us, and we stay with cells. Okay. Anna Osyczka from the Jagiellonian University. The floor is yours, and the topic is stem cells.
Yeah, it's not actually stem cells, but actually obtaining differentiated cells from easily available stem cells that we can get from adipose tissue. So what I want you to get interested, but before I, to the method we have now, and the method actually allows us to obtain osteoblastic cells quite easily, rapidly and effectively. But just to let you know why we are doing this. As you probably know, adipose-derived stem cells are now more commonly used in different therapies, but and they can be easy to get, much easier than, for example, taking stem cells from bone marrow. You can get really high volume of tissue and also a lot of stem cells from that. But in most clinical therapies that are using such cells, these cells stay undifferentiated. So basically, clinical therapies now use different properties of these cells, basically, just for now. So our idea was just to use them to obtain osteoblasts that are actually needed for several different situations when bone regeneration is actually required, and it is welcomes basically supports from any cells that can speed up this regeneration. And what's the problem with obtaining osteoblasts from adipose tissue? General procedure is quite long, so it takes like 2-3 weeks in culture to get a really osteoblastic phenotype in culture. So the idea, our idea, was basically to get them as fast as possible. And the method is actually shown in here. It's pretty simple and feasible in any clinical, preclinical setting. And basically, we have here just 3 major components. It's just fluctuating, so I don't know whether you can see. Basically, we have a surface that is a polymer ceramic surface, is bioactive. We have checked it for also some sort of bioactivity on its own, at inducing this osteogenic phenotype. It's also modified with some metal oxides, mostly zinc and strontium. Then we have a cocktail, which is shown in here. It's just not so well. It's fluctuating. Okay. So that's the cocktail. And basically, the cocktail contains 2 different inhibitors and recombinant human bone morphogenetic 2. And then there's this horizontal laboratory shaker, which is basically allowing us to mix the medium. Although the force we apply is just shear stress. So all this can actually provide us with osteoblastic cells very fast. And just to show you a couple examples, that's just 3-day treatment with the cocktail, with horizontal shaking. And not going into details, we get just 2 pretty late markers of osteoblasts as quickly as by day 3 culture after seeding cells. What else? We can also repeat this protocol, and you can see that we repeated it 3 times here, just added in between also just to enhance the whole process, recombinant BMP2. And what's also important, we have a way that after approximately 7 days, we can switch those cells to any other surface. The surface we use is no longer required. Cells maintain the phenotype and actually can differentiate, and is shown here, into really mineralizing osteoblasts. And mineral level you have on the right, so you can see that really is kind of impressive enhancement of expression of typical osteoblast phenotype. So to sum up, we can obtain osteoblastic cells or really mineralizing osteoblasts very quickly in culture. We can actually provide them, take them from any surface, any our surface. We have switch them to any surface any clinician or veterinarian clinician may need. Put them on the surface just to enhance bone regeneration. So cells can be actually provided in solution on a hydrogel or any biomaterial that is clinically used already. So we don't really need to provide it on our surface. And as quickly as 3 days after obtaining cells from fat tissue, we can actually apply this, and we have verified this so far in human cells. We are working now also and trying our solution in different animal cells for also veterinarian applications. The whole method has been developed actually by a group of people. Here you can see our team. That's my doctoral student on the left. On the right, there are 2 colleagues from University of Science and Technology, AGH or AGH in Kraków. So they are helping us with the surfaces, providing surfaces for this initial growth of cells. So basically where we are now, trying to develop this method into more advanced and more focused on specific clinical needs. So we are looking for any clinicians, clinical veterinarians struggling with specific, I'm saying specific bone regeneration cases when such therapy can be actually, when cells provided on any clinically available material will enhance this bone regeneration or difficult bone regeneration. Of course, once we switch to such focused cases, we will also look for business partners who will be helping us with developing commercial products and standardize the protocols. So thank you very much. That's it from me. If you want to talk, of course, I'm welcome any questions and discussion later on. Thank you.
Thank you so much. That's an excellent candidate, perhaps, to be matched with biomaterial research that we have seen before. So we look forward to fostering some collaborations. And now, the company again, a large one this time, EPAM Systems, and correct me if I mispronounce your name, but Anton Siomchen is on the stage. And we are, again, with IT for drug discovery, more focused on chemistry this time. So the floor is yours.
Hi everyone, so I'm representing EPAM System, which is a global company delivering software solutions for a lot of companies, including healthcare providers, big pharma, biotech, and we have dedicated LifeScience department, which I'm part of. And the topic of today's presentation is related to our LifeScience department. So whether you are organic chemist, any chemist, just student, or for example, chemoinformatician, sooner or later you would want to draw structures in the computer. And here one of the EPAM's open source solutions could help you. And here is a Catcher. So Catcher is open source and free software, which you could use from browser for editing small molecules, macromolecules, and reactions. It really looks modern. It has a great performance, very portable, so you could just go to the link and you have fully functional Catcher. So you could use it as it, or you could use it as a part of like larger program, because it's also available as a React and Streamlit components. So it's easy to integrate, for example, as an entry point for molecular prediction system or like any chemical system like ELNs. And Catcher is built with end users in mind. So whether it's a laboratory scientist who wants to share the structures with the colleagues, or just a student who wants to draw the structures to better understand how the chemical or biochemical systems works, or the academic researcher who wants to have publication-ready structures, Catcher has you covered. So a little bit of history. Development of Catcher started in 2006 under the GJ software, and in 2009 it became open source. So it means that now Catcher is also built by community, and the Catcher is a result of collaboration as it, because EPAM maintains a team of 10 developers which are working only on a Catcher, so implementing new features, or fixing some bugs which may occur. But as it's open source, any person could use it, then go to the GitHub repository and, for example, suggest a new feature to be added or fix some bugs in the software. And then in 2014, the GJA software solutions were acquired by EPAM. And for the next 6 years, Catcher was already used in many projects, but like not the paid ones. And starting from 2020, companies, like big pharma ones, could actually pay EPAM to implement new features, but it's not like they are paying and receiving their own version of Catcher. Sooner or later, these changes paid by big companies are available in the open source, so any company, any person could use the improvements for which big pharma paid. And also, last year, the support for macromolecules was added, so you could also not only use it as a small molecule editor, but for the macromolecules. If you want to create reactions, which is fairly common for organic synthesis publications, Catcher has a reaction layout to nicely align all your structures on the picture. Also, if you want to be sure which atoms from the substrates are in product, you could use reaction mapping, also in automated version. So just one click and you have the atom indices available in the drawing. And also you could add all sorts of information, catalyst info, conditions, and overall any text, any images could be added to the drawing and then exported. And reactions could be exported to reaction-specific formats, including smarts. So you could, for example, then do some work on these resulting smarts. Regarding small molecules, you could change most of the properties, so bond properties, atom properties. From the stereo point, you could use like normal stereo labels like EZ, SR, but also enhanced stereochemistry, like and in OR groups. And when you're ready with your drawing, you could export it using American Chemical Society drawing style, so your molecules would be nice and crisp. If you want to use other formats rather than like simple drawing, you could use widely used formats like mol files, SDFs, or as I mentioned, SMARTs or SMILES representation. If you're working with macromolecules, except for the mol files, you could also use FASTA, or if you want working with some complex structures, like for example, macromolecules with some small molecules attached, you also could use a Helm format for that. To make the drawing faster and simpler, you also have the library available of the small components like templates, functional groups, or salt. And this library could be customizable. New structures could be added to them to streamline your work. And if you're working with macromolecules, also monomer library is available for the peptides, so amino acids, as well as for presets and like building blocks for the RNA, like sugars and bases. Catcher's modality agnostic nature allows you to have everything in one picture. So, for example, you could have a peptide together with a small molecule attached to it, all on one single drawing, and then export it and use it in your publication report or anything. If you're working with macromolecules, especially with RNA, except for the building blocks like single bases, phosphates, or bases, you could also use like pre-assembled blocks. Without adding like single component, just add a pre-assembled set of base and phosphate, and you're ready to go. if you're working with proteins, you could also change the layout. So you could use a vanilla sequence mode, which is similar to text editor, but you could also use nice and colorful flex mode where all the connections are shown and different amino acid has different colors. On top of that, Catcher also uses Mu, which is EPAM's 3D molecular viewer, which is available straight from the Catcher. So in Catcher, you have everything is in 2D, but with click of a button, you could use 3D representations. If you have, for example, some data from patent, EPAM also has you covered because you could use Imago OCR, which is our toolkit for structure image recognition. So you could have your structures from a patent straight in Catcher and then modify them or export them to a better format. And for all the chemical functionality, Catcher uses Indigo toolkit, which in spirit is similar to RDKit, if you know it. So you could also use substructure search or fingerprinting thanks to the Indigo. And that's it. If you have any questions, you could... so it's all information. That was interesting. But yeah, you could just find me on LinkedIn or via email or on the LSOS platform here. And you could check what? Oh, I had different slides. So actually, it was supposed to be another QR code with a link to the newest version of a Catcher, but it's old presentation, so it's not there. But yeah, if you want to use Catcher, just go to the GitHub or our page and just Google EPAM Catcher, and you would have access to that. Thank you.
Thanks a lot. I hope this event brings more users to this open source tool, which is so nice and I'm sure useful to many of us. And last but not least, again, correct me if I mispronounce your name, but Zaid Jaber. Yes. Is it good enough? Okay.
Yes.
Will tell us a slightly different story on drug registration with the cultural context. So the floor is yours.
Hello, everyone. I'm Zaid Jaber, pharmacist and IP specialist from KG Legal. Today, our topic is about cultural and religious challenges to register a product, even pharma product or biotech product in Islamic countries. And it's critical for companies that want to expand to these countries. The Islamic countries' market size, it reached 108 billion in 2022, and it's expected to reach 142 billions by 2027. The Middle East and Africa region, it's worth 32.6 billions, with the largest market in Saudi Arabia, and the third largest market is United Arab Emirates. But one of the challenges to enter this market is to have a halal certificate. And halal term, it means the permitted or allowed, it's an Arabic term. And to have this certificate, your product should be free of prohibited materials like material from pig, blood, or ethanol. To obtain this certificate, it's granted for each product independently. So if you have this certificate for one product, it's not enough to enter the market with all products. So each product should have a different certificate. And this certificate has an expiry date, usually from 2 to 4 years, and you should apply for renewal, such like the ISO certificate and the accreditation for the manufacturing site. So a good question that is this certificate the same in all Islamic countries, or you should have a different certificate for each country? Generally, you have two main ways to obtain this certificate. The first is to apply to the Food and Drug Administration or the Ministry of Health in this country, or to have a halal certificate from accredited halal body. In Poland, we have like three main halal bodies. The first one is Polish Institution of Halal, and it's accredited in Saudi Arabia, Emirates, and Qatar. Halal Correct Poland, it's accredited in Saudi Arabia and Emirates. And Muslim Religious Union in Poland, it's accredited only in Qatar. There are many excipients used, and they may be prohibited or problematic in Islamic countries. First one is gelatin. Gelatin, it's widely used in the capsule shell, soft gelatin capsule and hard gelatin capsule. It can be from pig or from cow. If it's from cow, it will be allowed, but if it's from pig, it will be prohibited and you cannot enter the market. The second one is ethanol, and ethanol also it's prohibited, but it's less problematic if it's in granulation solution in the solid manufacturing. But it's more problematic if you used it as a co-solvent for liquid dosage form. But we have an example of bronchicum. Bronchicum, it's anti-cough medication, and it's contain co-solvent as an extraction solvent. It's contain ethanol, but it's permitted in many Islamic countries because the amount used, it's very minimal, and it was critical to extract the active material. Also, there are some active ingredients and the finished products that can be prohibited. The main one, it's heparin, because it can be from biosource, and the wide use, it's an anticoagulant, Clexane. It's from pig, but it's allowed in many Arabic countries and many Islamic countries because of the cause of emergency. However, there's an alternative now, it's Arextra, and it's chemically synthesized. So in the next few years, Clexane can maybe withdraw from the market due to the availability of the alternative. Also, some vaccines that contain blood materials or materials from pig also can be prohibited, especially vaccines that use trypsin, which is a material that an enzyme conversion from polypeptide to peptide. So if this enzyme from pig source, it will be prohibited. So the vaccine at all can be prohibited to enter this market. Other formal requirements is the Certificate of Pharmaceutical Product and Manufacturing Site Accreditation, which is almost unified in all the world. But for European company, if they have Certificate of Pharmaceutical Product, they can easily enter this market because some countries, they make a waiver for inspection for the manufacturing site if the product has a CPP from the European Medicine Agency. So it can easiest the way to enter this market. In KG Legal, we are specialized in pharmaceutical law, also in cross-border agreements, and we have partners from these countries. So if you are interested to expand your product, make some agreements, contract manufacturing, etc. in these countries, or want to enter this country and want to know more about the regulatory, we can help you in that. This is our LSOS platform, and I will be here tomorrow to discuss, collaborate, etc. Have a nice night, and thank you.
Thank you so much, and thanks for the patience and the great timing of the speakers. It was my fault that we are running late, so apologies for that. But the good news is that you don't need to wait until tomorrow for networking, because there is still some catering and coffee upstairs on the second floor. So if you are not in a hurry, you are more than welcome to go there. And we are going to have an inspiration night event this evening. It's by invitation apparently, but feel free to go there and give it a try. You can claim Sano and my name at the door. And tomorrow we are going to start with a startup session in the morning. Then there will be a very nice and hopefully inspiring discussion on the digital healthcare about noon. And then the final closing session, questions that matter, run by Kazek Murzyn, and finally a lunch. So this will be the end of the this year's edition of LSOS. But meanwhile, please enjoy the evening and see you tomorrow.
There is a place in the center of Europe where everything is possible. And every moment can mean everything. We will take you on a journey you will never forget, where imagination meets science and entrepreneurial creativity. Unique, our Kraków experience.